All right, team, here we are back again. Back in the energy photosynthesis and cellular respiration PowerPoint. We pretty much finished photosynthesis. I did want to just go back to this one slide real quick. Ah, shoot, went too far, sorry. Yeah, I wanted to go back to this one slide because it might be a little confusing. Darn it. Sorry. Doing the same thing over and over. Okay, let's try that one more time. Try not to click on it. Okay, so, we did this, and when we did it, we showed glucose going around. I'm sorry, we did use 6, right? We said that we needed to use a 6 because each time this cycle goes around, it takes 1 carbon dioxide in, but there's 6 carbon dioxides in glucose. So I just want to explain the image that's in your PowerPoint a little bit. They're using a 3 instead of a 6. Well, how can they show glucose coming out? What they're doing is they're just showing one of these glyceraldehyde 3 phosphates coming out. And then right here, they put 1 G3P. I guess that's from prior. And then they're putting those 2 together, and that would be the 6 carbons that would go on to make the glucose. So this can be a little confusing because they're going around 3 times. We went around 6, and that's why the numbers are different. Okay, just be aware. Okay, now we're going to get into cellular respiration. And you can see the basics here. One of the things to remember is that it's just the basically the complementary reaction of photosynthesis, okay? So what is really going on in cellular respiration? What's the big picture of it? Let's think about that. The big picture is that the energy in this glucose molecule is ending up in this other molecule called ATP. And it turns out that it's a 1 to 36 ratio. In other words, for every 1 glucose that you run through cellular respiration, you get 36 ATPs, 1 to 36. Okay, that's the net gain of ATPs per glucose. The other thing, this equation isn't balanced. There should be a 6 here and a 6 here and a 6 here, and that way everything adds up. And again, you'll notice, if we're going this way, we're doing photosynthesis. So it's the complementary reaction. We said how plants go round and round and do both, right? Okay, so you'll note it's a chemical energy to chemical energy transfer. One of the things that we know about cellular respiration is that this transfer is not very efficient. It's only about 20% efficient. And what I mean by that is we're talking about chemical energy. Only about 20% of the energy that was in the glucose ends up in those 36 ATPs. And so now we have this little thing that we have to answer, and the little thing we have to answer is, where's the rest of the energy, right? Because we know energy can't be created or destroyed. So what's going on there? Well, we think that the other 80%, or we know that the other 80% is liberated as heat. It's basically lost, but this is why we're able to stay warm. We have a strong heart that can circulate that heat in the blood, and this is why it's the heat of respiration, and that's what keeps our bodies warm. So I don't know about you, but I like to be warm. I'm not going to complain about this inefficiency. However, you know, suppose you made a motor or a heater and you took it to a trade show and someone's like, oh, that's a cool looking motor. How efficient is it? And you said, oh, it's 20%. They would laugh at you, right? So although 20% is not very efficient, it is the system that we are sort of stuck with in living things, okay? All right, so let's look at it. We break it down just like we did in photosynthesis into parts. And here are the 4 parts of cellular respiration, or steps, if you will. And so you want to know, what do we want to know about each step? We want to know what went in and what came out. And we want to follow the energy because, you know, when we start with glucose and oxygen, we're going to end up hopefully with 36 ATPs and some carbon dioxide and some water, right? Those would be the products. So first we have glycolysis. Glycolysis literally means to split glucose, and we'll see how that works. The oxidation of pyruvate, which is kind of an annoying little step that's necessary. The Krebs cycle, not to be confused with the Calvin cycle. And then the last step is called the electron transport chain. Let's see how this works. So glucose or glycolysis literally means split glucose. Okay, so let me blow this up just a little bit. That's not going to work. Hopefully you can see this okay. What is going on in glycolysis? Well, you can see we start with glucose. Here's glucose right here. Okay, and you can see the very first thing that happens is we're converting, it says 2 ATP are being converted to 2 ADP. So we're using some ATP here, we're releasing energy, right? And so you might be wondering, well, I thought the whole point of this was to store ATP or store energy in ATP, and here we are using it right here at the very beginning. We're using up ATP. Well, what's the mantra about money? What do they say about money? You got to have money to make money, right? Same thing here. We got to put a little energy into the system. And this energy we're putting in, it's actually called activation energy. It's what gets the ball rolling, so to speak. And so we're going to reap the benefits of putting this energy of activation in at the beginning, okay? So what this energy does is it takes that 6-carbon glucose and splits it into 2 pieces. Each of these P-galls are 3-carbon molecules. And that should make sense because glucose is a 6-carbon molecule. So you split it down the middle. Now you got 2 pieces. And now look what we do. We take some NAD plus, we convert it to NADH, we take some ADP and we convert it to ATP. We get 2 ATPs and an NADH. Now, think about it. This is happening twice, right? Per glucose. We're trying to keep track of the energy per glucose. And because we split the glucose into 2 pieces, this happens to 1 P-gall, and this happens again to the other P-gall. And then what we're left with are 2 pyruvic acid molecules. Okay, so for glycolysis, let's think about what we started with and what we ended with. We started with 1 glucose. We ended up with 2 pyruvic acids, or pyruvates, same thing, okay? And what about the energy? Well, we have 2 NADHs here, so we'll write, okay, we stored 2 NADHs worth, and we have 2 ATPs and 2 more, so we have 4 ATPs, but we used 2 of them up here, right? So we have to subtract those 2 out, and then we have 2 ATPs. Okay. Okay, so let's move on to the oxidation of pyruvate. Now, what I want you to realize is at this point, everything that we do kind of has to happen twice because if we're worried about the energy per glucose, well, we split the glucose into two pieces, and you'll see what I mean in a second. Okay, oops. Here we have this step called the oxidation of pyruvate. Pyruvic acid and pyruvate are the same molecule. So here's the three carbons I told you about, 1, 2, 3, right? Now look what happens in the oxidation of pyruvate. We have this 3-carbon molecule. We lose a carbon in the form of carbon dioxide right here. Okay, so we're losing a CO2. Now we have a 2-carbon molecule. And in the process of breaking that CO2 off, we're breaking a bond. We can use that energy to store some more NADH. And then there's this enzyme. It's called coenzyme A, and it combines with what we're left with when we lose the CO2 is an acetyl molecule. It's a 2-carbon molecule. It combines with this coenzyme called coenzyme A. And so then what we end up with at the end of the oxidation of pyruvate is a molecule called acetyl coenzyme A. And there's the 1, 2 carbons. Okay. Now, how many times does this have to happen, this oxidation of pyruvate per glucose? 2 times, right? So what do we start with? Pyruvate. What do we end with? Acetyl coenzyme A. And what about the energy? Well, we got an NADH here, but it happens twice per glucose. So the energy ends up being 2 NADHs. Okay. I'm going to do a little chart before we get too much farther so that we can keep track of the energy. And we'll keep coming back to this little chart, but I'm going to have to get out of this real quick. Okay. And before we get any further, let's do this. We're keeping track. We need to be able to show that glucose, right? So we need to be able to show that glucose turning into those 36 ATPs. So let's keep track. Glycolysis. What kind of energy did we get? We got 2 ATPs was the net gain and 2 NADHs. Oxidation of pyruvate, we got 2 NADHs. We're going to come back to this chart once we finish the Krebs cycle. Okay. So we'll be back looking at this in just a second. Okay. Now we have the Krebs cycle. And I can blow it up like that. That's good. Now you can see it. And see if it'll still annotate. Looks like it will. Okay, so this looks complicated, and it is complicated. Okay. One of the things they're showing is the oxidation of pyruvate right here, right? There's a pyruvate, oops, I wonder what happened. There's pyruvate, it loses the CO2, there's your NADH being made, and there's your acetyl coenzyme A. So what goes into the cycle is acetyl coenzyme A, which is, we said, a 2-carbon molecule. Off to the side here, I'm going to simplify this because I don't really care that you know the names of all of these molecules. It's not really that relevant to me. I will tell you in your reading, you might find this called Krebs cycle or the citric acid cycle, because citrate or citric acid is the first molecule that's formed in this process. Okay, so here's the deal. Let's just keep track of the carbons and the energy. Let's not worry so much about the names of these molecules. So if we start out with OAA, I'll just use the abbreviation, oxaloacetate. It's a 4-carbon molecule. So look how we can simplify this. We have a 4-carbon molecule. Acetyl coenzyme A comes in, it's a 2-carbon, right? Right here. That leaves us with a 6-carbon. And look what we do. We take the 6-carbon, we lose the CO2, and we get a 5-carbon right here, ketoglutarate. We lose another CO2. We get a 4-carbon. Let's show the CO2 coming off. Okay. And then 4-carbon to 4-carbon. Look, we're back where we started. Okay. So that's really very simple if you think about it. We're taking the 6-carbon molecule, we break a CO2 off, we got a 5-carbon, we break another CO2, we got a 4-carbon, and then around it goes through this sort of rearrangement back to oxaloacetate, and we're back where we started. Well, what about the energy? Well, check it out. From the 6-carbon to the 5-carbon, we're able to store, you see NAD plus becoming NADH right here. There's an NADH. From the 5-carbon to the 4-carbon, we do the same thing. We store some more NADH. Let's draw that one, NAD plus NADH. Okay. And then from this 4-carbon through this rearrangement, look what we do. They say GTP, it's really just ATP, it's all good. So ADP is going to ATP. So there's some more energy stored. Now we hadn't seen this FADH2 molecule yet, but it gets stored. FAD plus FADH2, and then one more right here between malate and oxaloacetate, we get to make another NADH. So this is nice because it shows you the energy, right? And so each time the Krebs cycle goes around, we go from 6-carbon to 5-carbon to 4-carbon, and then we rearrange back to oxaloacetate. And what about the energy? Well, how many NADHs and how many times does the Krebs cycle have to go around per glucose? Twice, right? So really all of these are going to, per glucose, this is all going to happen twice. So if we add up the NADHs on the first turn, we got 1, 2, 3, and then we would turn again for the second acetyl coenzyme A, and that would be 3 more. So we got 6 NADHs, right? FADH2, well, 1, 2 right here, okay? So 2 FADH2s, and then ATPs, we get 1 here, and it goes around again for the second acetyl coenzyme A, and we get 2. Okay? So now at this point, we say all of the energy that was in the glucose has been systematically transferred to NADH, FADH2, or ATP, or it's been lost as heat. Okay? So let's go back now to our, let's go back now to our little chart, and let's put these in there for the Krebs cycle. And we're going to say, okay, we got 6 NADHs per glucose in the Krebs, 2 FADH2s, and 2 ATPs. Okay. Now we are back where we're going to say, okay, all the energy that was in the glucose is now we've accounted for it all here, or it's been lost as heat. Okay. Okay, what about the electron transport chain? What's that all about? Let's see. Oops, went the wrong way. Here we go. We're going to just draw this. This shows the electron transport chain with all the molecules associated with it. It's a little bit small as an image, a little bit hard to see maybe. So we're going to draw it out on our own. And, we already saw an electron transport chain. We saw an electron transport chain in photosynthesis. Remember, we said the electrons get passed from a higher energy to a lower energy. Okay, so let's see how that works. Let's go back to our other screen. And it looks like I'll have just enough room, got plenty of room here to draw. Okay, so here's how it works. I have a series of steps like this. We're going to call this the ETC, the electron transport chain. And it turns out that what's donating the electrons to the electron transport chain is either NADH or FADH2. The whole point of the electron transport chain is literally to convert the energy from NADH, FADH2 to ATP. We're trying to end up with just ATP. Remember, in the very beginning of our discussion, we talked about how NADH and FADH2 were kind of intermediary molecules between glucose and ATP. So we're trying to get that energy to ATP. And remember, what happens here is the electrons get donated with a hydrogen, and then what we're left with is NAD+. Where's it going? Well, it's going to pick up another H in the Krebs cycle or glycolysis or wherever it was made. Down the steps come the electrons with their hydrogen, right? And waiting at the bottom of the steps is oxygen, and it snatches one hydrogen, it snatches another, and you make water, which is, remember, a product of cellular respiration. In the process of going from this higher energy to lower energy, we are then actually able to make ATP. So right here, we're turning ADP into ATP. It's like an ATP making factory. There's a rule about how many ATPs you get. The rule says, and this is a little frustrating, but you just have to learn it. If it was NADH that donated its electrons, you get 3 ATPs out of the deal. But if it was FADH2, you only get 2 ATPs. So you just have to remember that. You just have to kind of commit that to memory. So just to reiterate, what is the electron transport chain? It's a way to convert the energy of these molecules into ATP. These molecules donate their electrons, down they go. We get water ultimately at the end as a product. And you can see the rule is if it's NADH, we get 3 ATPs out of each NADH, and we only get 2 if it's FADH2. Okay. So here I've shown NADH donating the electrons, so I'm going to put 3 here. Okay. So now, guess what we can do? Now we can add up all of the ATPs that we got. And as we said, we're supposed to get 36 ATPs. So let's see if we got it or not. See what happens. Let's get rid of all this. And let's just kind of finish our chart out. Okay, so let's make a table now. Let's do this, because we want to know what the total is, okay? And ETC stands for electron transport chain. And we want to know what's the total ATPs that we're going to get. So if we start at glycolysis, well, 2 ATPs, you don't have to do anything to those. We just move them over here into the totals column. Okay. What about these 2 NADHs? Those have to go through the electron transport chain. And we said for each one, you're going to get 3 ATPs. So let's multiply each of those by 3, right? That should make sense because there's 2 of them and each one goes through the electron transport chain. We get 3 ATPs for each one, so that's 6 more ATPs. And so you can see we're adding up all the ATPs. Here's 2 more, got to run those through. So there's 6 more. Now here's 6 NADHs. Here we're really racking up a bunch of energy. There's 18 ATPs. Here's FADH2. We run it through the electron transport chain. Now we only get 2 ATPs, right? Because we said that's just the rule. If it's NADH, you get 3. If it's FADH2, you get 2. So 2 times 2 is 4. And then these 2, we just move over. They're good to go. And if we add these up, add them all up, and what do you get? 38 ATPs. We were only supposed to have a net gain of 36. Now we took into account the 2 that we used up in glycolysis because we only counted 2 here instead of the 4. We subtracted the 2 out that were the energy of activation, but we still have these 2 extra ATPs. And I'm about to explain to you why there's this discrepancy and what's going on. So here's the thing that I didn't tell you before. Glycolysis, this process occurs in the cytoplasm of the cell. The cytoplasm is just the liquid goo that surrounds all of the organelles. So glycolysis doesn't occur in an organelle, okay? The rest of this jazz all occurs in the mitochondria. So we take glucose, it's in the cytoplasm, we split it into 2 pieces, right? Two pyruvic acids. Well, guess what we have to do? The mitochondria has a membrane. We have to move those 2 pyruvic acids through a membrane into the mitochondria. Is that free? It is not free. It is going against a concentration gradient. And so guess what we have to do? Right here between the cytoplasm and the mitochondria, we got to subtract minus 2 ATPs. We're going to call this a shuttle cost. And it's quite literally the cost of doing business, the cost of moving those pyruvic acids into the mitochondria where the rest of this stuff happens. Okay. So then that's how we get our 36 and not 38. There's the net gain of ATPs, 36. Okay. So I know it's complicated. I know there's a lot of stuff going on, but you just have to, watch the video a couple of times. It'll start to make sense. Do your reading. What's the big picture? Well, we should be able now to account for each of the reactants and products, right? Heck, we don't even have to worry about balancing the equation if you don't want to at this point. You know how to balance it. Okay. Oops. We should be able to see where this stuff comes in and out though. Where did we see glucose go in? At the very first thing that happened was glucose went in glycolysis and it was split. This is the big picture, y'all. We had to go through all those nitpicky details in order for me to feel good about you understanding what exactly is going on. But at this point, step back away from all the details and look at the big picture and know something about the energy, okay? So where did we see oxygen? At the very end, the very last thing that happened, right, was oxygen receiving those hydrogens with their electrons and making water. So at the very end, the electron transport chain is where we saw oxygen being involved. Where did carbon dioxide come out? It came out in the oxidation of pyruvate and also the Krebs. Everybody take a deep breath and now exhale. You're exhaling carbon dioxide. You're respiring. Where did that carbon dioxide come from? Literally came from the process we just discussed. The oxidation of pyruvate in the Krebs cycle. This is occurring in every cell. Every living cell in your body is doing this all the time. Isn't that wild? And then where did we see water come out? It's the product that's formed at the very end of the electron transport chain. Okay? All right. Well, that, my friends, is cellular respiration, aerobic. A-E-R-O-B-I-C. What we just did is aerobic cellular respiration. Why aerobic? Because there's oxygen involved. What happens if you take the oxygen out of the equation? It becomes anaerobic respiration. And so the last little piece of info that we want to do in this video is we want to talk about anaerobic cellular respiration, okay? You're actually a little more familiar with this than you think, and we'll see why in a second, okay? So here's our drawing. These cytochromes are the electron acceptor molecules that shuttle those electrons down the electron transport chain, and there you can see there's oxygen and we made water. So let's just have a little reiteration of aerobic cellular respiration. Aerobic respiration, I don't know why it's spelled terribly wrong. Sorry about that. One glucose molecule that completes aerobic cellular respiration yields 36 ATP molecules. So it's a 1 to 36 exchange rate. This reaction is only about 20% efficient. The rest of that energy is not mysteriously vanished, right? It's released as heat. Okay. Let's look at anaerobic cellular respiration. And let's look at what's going on here. There are actually two different anaerobic pathways. Now, here's the thing, y'all. If there's no oxygen, then there's nothing to receive the electrons and the hydrogens at the very end of the electron transport chain. Think about the entire process of cellular respiration like a factory, right? And think about oxygen as the guy or the girl at the end of the assembly line in the factory that takes the product and puts it in the box, okay? If that person walks out of the factory, everything gets backed up, doesn't it? It gets backed up. And so if you take oxygen out of the equation, then there's nothing to receive those electrons and everything just sort of gets backed up and everything shuts down. When the electron transport chain shuts down, you run out of NAD plus to use in the Krebs cycle. And when you run out of NAD plus, the Krebs cycle shuts down. And ultimately, everything just kind of shuts down, okay? So there is an alternative pathway for organisms to do where you don't need oxygen, okay? And it's called anaerobic respiration. Now, it starts out the same. It starts out with glycolysis, and we get our pyruvic acids. So that doesn't change. I'm going to draw it over here in the sidebar. We start with the glucose. I swear that says glucose, I'm sorry. We do glycolysis. Okay, and then we're left with pyruvic acid. In the process of doing glycolysis, remember we got 2 ATPs and we got 2 NADHs. Now, here's the pathway. It looks like this. There's actually 2 different pathways okay? One of the pathways gets rid of a CO2. This is the one you're familiar with. And you're left with alcohol, which is a 2-carbon molecule. Remember, pyruvic acid is a 3-carbon. The other pathway does not get rid of a carbon dioxide, and you're left with lactic acid. Okay? But in both of these pathways, what actually happens is the NADH right here, this doesn't make a lot of sense, but this is what happens, gets converted to NAD plus again. So you're kind of dumping energy here. You're having to do this because you need to regenerate this NAD plus in order to keep glycolysis going. If you don't have NAD plus, you can't make NADH. And then glycolysis would shut down. So it's kind of a compromise where you're dumping energy. Same thing would happen on this side in this pathway. It's just you don't lose the CO2. You do still dump the energy. Okay, so what's the net gain of ATP for anaerobic cellular respiration? A measly 2 ATP, because we're using these up right here for nothing. We're basically just dumping energy. Note, lactic acid is a 3-carbon molecule, alcohol is a 2-carbon molecule. Why are you familiar with anaerobic respiration? Because it's glycolysis plus fermentation. And you're familiar with yeast fermentation if you've ever drinking a beer. What does a beer have? It has alcohol and it has bubbles, right? The bubbles are the CO2. There's your bubbles, and there's your alcohol. That's the part that makes you feel a little loopy, right? Okay. Lactic acid, you're also familiar with lactic acid fermentation if you've ever eaten something like yogurt or salami. This is beer, right? Or sauerkraut or kimchi, right? We use lactic acid bacteria to preserve things like dairy products or meats. So salami is basically fermented meat, okay? It's a way of preserving vegetables or dairy products or meats using lactic acid bacteria that generate lactic acid, which is a natural preservative. We do the same thing with alcohol, okay? Alcohol originally was used as a way to make water safe to drink. I mean, if you ever read books or watch movies about them took place in the Middle Ages, like King Arthur's time, they didn't ever say, and then we drank some water. What did they drink? Why didn't they drink the water? And what did they drink? They drank beer, mead, wine, lambic, right? Because the water they knew would give you a bellyache. But if you run the water through this fermentation process, it actually sterilizes it and makes it safe to drink. It kills the bad microbes that might be in the water. So, you know, more than likely, this was an accidental discovery. No one probably invented wine or beer. They probably just had some sugary fruit in an animal bladder maybe, and the natural yeast on the skin would have fermented the sugars and made an alcoholic beverage. Now, one thing to consider is the alcohol content would have been relatively low because of the wild yeast not being very tolerant to alcohol. It probably would have been about 1 to 2% alcohol rather than 5 to, you know, they have the high gravity beers now can be 10 or 11%. Okay. So anaerobic respiration is glycolysis plus fermentation, and there's 2 fermentation pathways. One is alcohol fermentation, one is lactic acid fermentation. They're both used and useful to humans. Okay. Well, gosh, y'all, we're doing so well. I think there's one more slide which says, yeah. The only thing it should say is molecules of what? It says 1 glucose molecule goes through anaerobic respiration, yields 2 molecules of ATP. So now we have this little discrepancy, don't we? Because if if you do aerobic respiration, you get 36 ATP. And if you do anaerobic, you only get 2, but energy can't be created or destroyed. So where's the 34 ATPs worth of energy? Has to be somewhere. We already accounted for the heat loss, so we can't say it's lost as heat. We're missing 34 ATPs worth of energy. Guess where it is? It's trapped in the product. So what's the product in this case of fermentation? It's trapped in either the lactic acid or the alcohol. What's the evidence of this? Well, if I had a bottle of Everclear, a handle, as my daughter calls them, of Everclear, which is pure grain alcohol, it used to be 180 when I was a kid, and now it's 150, I guess. But at any rate, if I had a handle of pure grain alcohol and I had you sitting in front of me, I popped the cap and I just start splashing it out into the audience, dumping on my head, and then I'm like, okay, anybody got a light? What are you going to say? You're going to hopefully go run to the president of the college and be like, this guy's crazy. Why aren't you going to give me a light? What do you know about alcohol? It's flammable, right? So what we know is that energy that's trapped in the alcohol has potential energy, and the evidence of that is that it's flammable. So if I ask you, if aerobic cellular respiration gives you 36 ATPs and anaerobic only gives you 2, where's the rest of the energy? It's in the product. It's in the alcohol or the lactic acid. Got it? Got it. Okay, team. That's a great video. I hope you've enjoyed it. You can watch it over and over. If you don't understand, feel free to check in with me. Again, always going back to the big picture being the general chemical equation. Okay, so try to make sense of all this, and we will see you next time.